Hi everyone, I'm Corey Ratcliffe, a PhD student in Simon Keller's Liverpool Brain Lab. That's brain research using advanced imaging in neurology. I'm going to give you all a brief overview of my recent work, which has involved using two AI-driven software to create 3D images from 2D magnetic resonance imaging scans, the latter of which being representative of clinically acquired MRI scans. After this, I evaluated the ability of these synthetic images to identify potential atrophy within the subcortical structures of patients with idiopathic generalized epilepsy. Most people watching this will be familiar with MRI terminology, but as I will be referring to 3D and 2D MRI throughout the talk, I'll just clarify what I mean by this. When I say 2D, I'm referring to images that are still 3D, that is, still stacks of 2D slices. But whilst 3D MRI scans are made up of voxels that are isotropic, i.e. 1 millimeter by 1 millimeter by 1 millimeter, a researcher or clinician may choose a 2D sequence to cut down on scan times by reducing the number of slices, thus elongating the voxels in one direction to something like 0.4 millimeters by 0.4 millimeters by 3 millimeters. The in-slice resolution is still high, but when looked at it sideways, the resolution drops. So quantitative analysis of MRI can leverage increased sensitivity and group-based designs to capture information that qualitative evaluation may not be able to detect, which has been particularly informative in epilepsy research. Atrophy, or abnormal volumes, are common findings in the brains of individuals with epilepsy, both of the cortex and in select subcortical structures such as the hippocampi and the thalami. Malrotation of the hippocampus in juvenile myoclonic epilepsy is suggestive of a genetic imaging phenotype, and other syndrome-specific imaging biomarkers have been identified. Links between epilepsy-related cognitive impairment and structural imaging abnormalities have also been established, and the putative implications for prognosis and surgical planning are significant, even if implementation is at an early stage right now. One reliable finding in idiopathic generalized epilepsy, that's IGE, is subcortical surface deformation, potentially indicative of atrophy of the hippocampus and the thalamus, for example. This is problematic, however, because so much of the data acquired in routine care uses contrast enhancement, is artifacted, missing metadata, or is recorded in 2D, which is acceptable for qualitative evaluation, but unsuitable for use with many of the software packages used by neuroimagers. On the right are two scans that I've used in my study. The top one is a gold standard 3D MRI, the bottom a low-resolution 2D scan of the same participant. Trying to work with 2D images can result in unreliable quantitative estimations or even pipeline-ending errors. For these reasons, a lot of clinically acquired data is neglected or considered unworkable for quantitative analyses. This means that there is a huge amount of MRI data required that neuroimagers can't really use in its present state. With a view to this, two software have recently been developed for use With a view to this, two software have recently been developed that use AI to estimate research quality scans from otherwise unsuitable data. These are SynthSR and DL+DiReCT, and the aim of this study was to see how accurately these two software could synthesize 3D data from a 2D analog. This was a case control study for which 31 individuals with IgE were recruited and 39 healthy controls. The two groups were comparable for age and sex distribution, and they both underwent a scanning session in a 3 Tesla MRI at the Walton Center NHS Foundation Trust, during which a 2D and 3D structural MRI scan were required for each participant. The latter provided us with our ground truth. So using DL+DiReCT and SynthSR separately, the 2D scans were synthesized into 2 further images per participant, bringing the total to 4 images per subject. That's a 3D scan, a 2D scan, a DL+DiReCT created 3D image, and a SynthSR created 3D image. The 4 images were then pre-processed in parallel. Free surface reconstruction pipeline was used to extract volume and thickness measurements for 181 predefined cortical and subcortical regions, as well as to provide isolated segmentation images of each region. FSLNet was used to estimate the surfaces of 14 subcortical structures: the bilateral nucleus accumbens, amygdalae, caudate, hippocampi, pallidums, putamen, and thalami. The brainstem segmentation was omitted due to poor The brainstem segmentation was omitted due to poor estimation historically. The initial comparisons were performed within subjects, so the sample was not split. The first analysis I did was correlate the volumes and thicknesses taken from the 2D and synthesized images for each region with those taken from the 3D scans. I then computed the dice coefficient for each region from the 2D and synthesized images with its analog in the 3D dataset. If you're unfamiliar with the dice coefficient, it's effectively a measurement of spatial overlap. An example is shown on the right that highlights how two similar appearing structures can only be minimally overlapping. The intra-class correlation was next, which measures the similarity between two datasets. And finally, the sample was split into cohort and control, and the two groups were compared on the basis of their subcortical shapes. If an inwards deviation in the shape of structure is found, this can be suggestive of regional atrophy that might be too small to register in whole structure comparisons. Again, there is a diagram on the right that offers a simplified example of what we might be looking for. Firstly, correlations were high overall. This was mostly done to check that there had been no catastrophic biases present, but unfortunately, it doesn't really tell us a huge amount about the relationship. Dice coefficients were much lower overall. Spatial overlap between regions of the cortex in the 2D and synthesized images averaged around 16%, but did improve to 26% in the subcortical structures. Importantly, spatial correspondence was no higher between the 3D scans and the synthesized images than the 3D and the 2D scans. The 2D scans were the highest overall, albeit only marginally. So to evaluate the use of synthesized images in volumetry, and to see how much or how little the 2D and synthesized image metrics differed from the 3D scans, intra-class correlation coefficients were computed for the cortical region volumes, subcortical region volumes, and cortical grey matter thicknesses. Each line on this series of charts represents a trend line of the correlation between the 3D region and one of its analogues, with perfect correspondence represented by the diagonal. If you look at the overall patterns, you can see again that there is little difference between the 2D scans on the left, the DL+DiReCT images in the middle, and the SynthSR images on the right, although volumes were better estimated than grey matter thickness globally. This is where we split the group into controls and individuals with epilepsy. Here you can see the subcortical structures, with inwards deflation of their surfaces, as measured in the 4 different images, represented by the colours in the legend. Anywhere that is blue represents a potential spot of atrophy in our idiopathic generalized epilepsy cohort, as measured using the raw 3D volumes, that would be the 3D scans. Reaching statistical significance were the left accumbens, the right caudate, the bilateral putamen, and the right thalamus. The 2D scans were not sensitive to inward surface deflation when compared to our ground truth. As you can see, only the right hippocampus is here as a trending result and did not reach significance. And the two synthesized datasets picked up on more of the true positive results, although neither found all of them, and both were vulnerable to false negative results, such as the left hippocampus in the SynthSR. No. So what does this all mean then? Well, 3D images synthesized from 2D MRI scans offer no tangible benefit over 2D scans for volume and thickness analyses. However, they may be more sensitive to smaller deviations in 2D data at the cost of requiring careful interpretation and control. In anticipation of a potential question, I didn't train my own models, so I can't comment on the accuracy of these methods when used with custom training sets. But overall, synthesized images in quantitative neuroimaging should be approached with caution. But if used sensibly, they can be used to coerce 2D images into software-compatible 3D images, reduce the resource burden of pilot studies, and allow for the quantitative analysis of retrospective clinical data, which would otherwise be ignored. That's it from me. My poster is number 875. Please feel free to stop by between 12:15 and 2:15 on Sunday the 23rd or Monday the 24th if you'd like to discuss anything I've talked about, or scan the QR code to get in touch with me, see all the references. Thanks to all my co-authors and lab mates, and thank you for listening.